Hi, now we want to review our Genetics 101 for molecular principles in BME. So let's think about the genetics and inheritance. So Greg Mendel's discovery of these principles of genetics was revolutionary. Even before hundreds of years ago, knowing about these molecular principles of the DNA and RNA. So that is a fascinating. The importance of the discovery of the Greg Mendel is not about the experiment itself and his interpretation, but the power of by bringing a theory of how this inheritance works and predict the future. That's the power. That's what we acknowledge him as the father of genetics. So the genetics is a study of heredity and hereditary variations. And we talk about gene a lot. And what is gene? Gene is a locatable region of this genomic sequence first. And so a unit of inheritance as a gene, which is a locatable one, in somewhere in the chromosome. And so these genes are located on the chromosome, and that's within the nucleus. And this double-stranded DNA is packaged with a histone protein to form a material called chromatin. And let me go through once again. So DNA packaging in the nucleus of a cell. So people say who work with DNA call DNA as very sticky. The reason is, remember that DNA is composed of this nucleotide with a pentose sugar molecule and phosphate and DNA base. This phosphate has PO. There's a lot of negative charge, so DNA is negatively charged, and this makes DNA to wrapping around histone. Here, this is a histone to form a nucleosome. And you can see here, nucleosome. And nucleosomes are further packed together to form a 30 nanometer chromatin fiber, and additional levels of packaging result in highly condensed chromosome structure. So think about this real dimension of this whole process. You know, in the past, we only use an optical microscope to see micron level, which is this chromosome. When it's metaphase, it looks like this. It's 1.4 micron, so people, humans, can see the shape. But DNA is so tiny. Can you see this? This diameter is about 2 nanometer, and that's DNA double helix actual size. And that's wrapping around a base. It's a histone protein, and this package into nucleosome about 30 nanometers, still far small. And even this will become a scaffold onto chromosome scaffold to form about 300 nanometer, which is about kind of a good microscope can barely see. And it condensed with this form, and we call as a coiled coil structure. This is just for reference, but to give you an idea that DNA double helix 2 nanometer, at the simplest level, this chromatin is double-stranded helical structure of DNA. And this is wrapping around histone proteins, which is composed of 8 histone molecules and about 1.6 times rotation on average. And with this, it forms a chromatosome and nucleosome structure forming about 30 nanometer. And that forms loops averaging about 300 nanometer. And with this, about 300 nanometer fibers and fold to this. And this coiling results in to this final chromosomal structure, which is visible with our microscope. So in the past, so we have some genetic problem at the level of chromosome, people can actually find out. A good example is Down syndrome you can recognize the people which is affected by this, and people look at their cellular nucleus and those chromosomes, and people realize this. There's a 21 chromosome, there's a not only 2 but 3, so we call it a trisomy. So this observation of human chromosome is called human karyotype. So with this, we can start to look at some of the genetic heritable diseases. So these chromosomes can be isolated from a cell, and we can display it in a karyotype. So it's very simple. You just look at them and then sort them based on the length. 1, 2, 3, 4, that's shorter order. And then, so you can find pairs of this chromosome. One's typically from father, the other from mother. Okay, so this is as a 20 totally this pair. And this genetic makeup of an individual we call genotype. But the observable traits in an individual is called phenotype. So genotype versus phenotype is a very important discussion in genetics. And there are still, we do not understand. Some people argue that every disease will be a genetic disease, which has its own foundation too. So this karyotype has 23 pairs of homologous chromosomes, and the nuclei of all these somatic cells. Somatic cell means in our body part in comparison with the germline cell, which is coming from our sex cells. Okay, so nuclei of all somatic cells will contain, you can see this 22 identical, while the sex chromosome in this case is a male, so it's XY. You know that for female, it's an XX. So these somatic cells contain 22 identical pairs of chromosomes plus a pair of sex chromosomes. And this chromosomal level aberrations, which can be observable with a microscope, we call it as a karyotype analysis. And for example, this number 12 chromosome, so one from father, one from mother, and these is a pair as a homologous pairs of chromosome, and the genes of one chromosome is called allele. So there are 2 alleles for the character, and the location of this on the chromosome of certain entity, certain genetic unit is called the gene, and that's a gene locus for that character. So this is a form for somatic cells, a diploid cell before meiosis with 2 pairs of chromosome, and we indicate, so this is a homologous chromosomes, one kind and the other kind. Let's consider one locus of the gene 1 and the other locus of gene 2. And this, once meiosis happens, that leads to 4 possible haploid cells. So that one from this chromosome and the other from this chromosome. This is 2 and 2, so 2 multiplied by 2 is 4 different kinds you can think of. So for example, this is a father actually from a sperm and mother's egg, which has this haploid cells. Then once it's fertilized egg, that will contain a combination of these 2. So you can so look at this homozygous at locus for, so these one and these one, you see the allele of one gene is different and we call it heterozygous, and these alleles of gene 2s are the same and we call it as homozygous. So the human cells that contain total 46 chromosomes, which is containing 2 copies of each chromosome, and that we call diploid. This di means a pair or 2. And the paired chromosomes are called homologous chromosomes. So in this one and this one. But the reproductive cells, such as sperm and eggs, will have half of this chromosome. And these cells we call a gamete. Okay, so sperm cells or egg cells, and so they contain only half of the number of chromosomes, and thus we call it as haploid. Okay, so germ cells are haploid cells, and the location of this gene, the physical location of a gene on a particular chromosome, we call it as a gene locus. So I want to summarize genetics in terminology, which is important for understanding for the rest of the class. So each heritable feature, such as eye color or hair color, and that we call as a character. And that character can have a variant. For example, eye color, I have a brown eye color, some people has a blue eye color, and those we call it as a trait. Okay, so each of alternative version of a gene as DNA sequence in the gene can vary from one chromosome from another because we have a copy of chromosome. So the genes can carry the same sequence or different. And each alternative version of this gene we call as a allele. Okay, so homologous chromosome is a pair of chromosomes with the same gene, although may possess either identical alleles or two different alleles. So when these alleles are identical individual, we call it as homozygous for the gene controlling that character. If they are not identical, then we call heterozygous. Okay, so these traits, such as a blue eye, is exhibited, we call it, because we can see it as a phenotype. However, the genetic makeup can be not the same. So that is a genotype. So with this, we have to discuss how the genotype and phenotype relation. So one question we can ask is, hey, I can see your trait. I have a brown eye color, brown hair, yellow skin color. Can you infer my genotype from phenotype? The answer could be not exactly, because maybe the phenotype is the same, but the genotype could be different, okay? So that's, we call it as individual with two different alleles called the heterozygous. One of the alleles could be dominant or fully expressive, and the other can be without noticeable effect, is called recessive. Because of these aspects, we cannot infer a genotype directly from phenotype, but we can actually think about the probability. So all of this lead to us the question of, why do we study this? Because many diseases are related to our genes. And so we can ask, hey, there are genetic diseases, and such as single gene defect. I told you cystic fibrosis, and this is affecting CFTR gene, and Huntington's disease. Still, we cannot cure these individuals. And what about cancer and cardiovascular disease or type 2 diabetes? And this we know a lot more than before now, which is involved with the polygenic, meaning by it's not just single gene defect, it's involved with changes and alterations of multiple genes. And that we are now considering this has a lot of effect on environmental aspects. So it's very hard to treat by these diseases. And so we have to know a little more about this. Okay, here comes a practical question, which I guess you have already practiced. but just to make sure you know the basis, calculate the probabilities for the outcome of mating a homozygous male having capital AA with a heterozygous female, capital A, and lower capital A. So, and capital A usually means dominant, and lowercase usually recessive. So the probabilities outcome of a genotype and phenotype. So we discuss molecular principles in DME. So we discuss the structure of protein, which is a polymers of amino acid. And these diversity of 20 amino acid structure influence the eventual resultant protein three-dimensional structure and its function. And regarding to genetics, we understand the structure of nucleic acids, and that's composed of polymers of nucleotides, and how this structure is different between DNA and RNA polymers. And the importance of the DNA and its structure directly leads to its function, which is storing genetic information. And that leads to our better understanding of genetics from the past as a molecular level. And that we call now as a genomics, because we now having information of the whole genome. So next time we will get into more detail. Thank you for your attention.